Our first speaker today in this room will be Professor David Nichols. David is an adjunct professor at the Eshelman School of Pharmacy at the University of North Carolina. He appears as author of more than 300 scientific publications. He initiated the founding of the Hefter Research Institute in 1993. And in addition to developing novel psychoactive substances, the Nichols lab also synthesized high-purity MDMA, DMT, psilocybin, and other substances for research programs. So it's really this lab that made it possible for psychedelic research in the modern era to happen, because not only the permission of authorities was needed, but also someone who would synthesize really high-quality, pure substances that could be used for scientific research. So with Professor David Nichols, we have one of the pioneers of modern psychedelic research here with us today. And he will be giving us a talk called Psychedelics as Medicines. And please give a warm welcome to David Nichols.
So I'm delighted to be here. I was very excited to be invited to this Psychedelic Psychiatry conference, and I have to say that the organizers' goal of having it be very professional and high quality has met the mark. I've been really impressed by the quality of the presentations, and this is really a different kind of meeting than I've often been to over the years for psychedelic science. So anyway, and I'm going to do something a little different. Usually I talk about some of the chemistry that I've done, and today I'm going to try to bring you up to speed and how I think things are happening in the brain, talk a little bit about neurophysiology, but not at a level to confuse people, because I recognize most of you are not highly trained in physiology or more clinically oriented, but we'll see how that goes. I hope I can convince you that we know a little bit about what's happening in the brain, and so let's get started. The background of this picture is by a visionary artist named Pablo Amaringo from the Amazon, and he paints pictures that portray the kinds of effects that people have or see, experience when they take ayahuasca. So I thought that would be an appropriate way to start off. So this is the definition that I've used for. A dog already doesn't like what I'm saying. This is a definition that I've used in my talks for many years, and it's from the 7th and 8th editions of Goodman and Gilman's Pharmacological Basis of Therapeutics, which is a big, thick book about like this. That's the quote-unquote pharmacologist Bible. So psychedelics are drugs that are unique because they induce or compel states of altered perception, thought, and feeling that are not or cannot be experienced otherwise except in dreams or at times of religious exaltation. If you were a pharmacologist, that definition would be really unique, because when you think about drugs for cardiovascular or GI problems, etc., they don't have a definition like this. And I'm going to show you some of the background that really led to this kind of a definition. So historically, this will be a review for many of you, and a couple of these slides are similar to slides I've seen in other talks, but let's just talk about where these came from. So in South America, the drug we know most about today is psilocybin. That's being used in most of the clinical studies. And they originally came from so-called magic mushrooms. These were used by the Aztecs. They were called teo nanacado, which means flesh or food of the gods. And if you analyze these, Albert Hofmann, who discovered LSD, determined that psilocybin was the active component in these mushrooms. So this is a four-substituted dimethyltryptamine. That RO is at what's called the 4 position. And if that R group is a phosphate, which is a phosphorus with 3 oxygens attached to it, it's a phosphoryl oxy group, and that's psilocybin. And that's actually what's called a prodrug, because when you take psilocybin in the body, that phosphate group is cleaved off enzymatically, and that R group becomes a hydrogen, and that's psilocin, which is the active species which actually acts at the receptors. Now, we know about something called soma. In the ancient Indian literature, in the Rig Vedas, there are hymns written in praise of soma. Nobody knows what soma was. It's been lost to antiquity. But there's some evidence it might have been a mushroom. So this is a embroidered woolen textile that was dug out of a tomb in China in 2009. And what this is a Zoroastrian, see if I can make this thing work. It's a Zoroastrian ceremony, and this priest here is holding a mushroom, and they suggest it looks like Psilocybe cubensis. Now, he wasn't holding that mushroom out as a dietary object. It must have had some sacred significance. And this is in the context of a ceremony. There's a fire down here. So it may well be that the soma of ancient India actually involved the use of psychedelic mushrooms. We know they grow worldwide, and they're very profound. So we drank soma, we became immortal, we came to the light, and we found gods. So this would be very typical of something that might happen after a psilocybin experience. Also, we had the Eleusinian Mysteries in a village called Eleusis, 15 miles outside of Athens. Every year in the fall, there was a secret ceremony. We call it the Eleusinian Mysteries. There was a building called the Telestrion, where adherents went, and you could participate. Any adult could participate if they hadn't been convicted of murder. And they drank something called kykeon. We don't know what it was. The only thing we know about it was it had barley and mint in it. So people have looked at that and said, well, barley, maybe it was infected with ergot, so maybe ergot alkaloids were in there. Well, it was a secret ceremony. You couldn't talk about it. If you did, they would kill you. So nobody knew anything about it. So why would you say barley was in there if there was something related to barley? So I think it was kind of just a cover. So here's a scene from the Eleusinian Mysteries. This is a bas-relief, and if I can make the pointer work, what we have here is a couple of mushrooms. A scene from the Eleusinian Mysteries with Persephone taking a mushroom for Demeter. This is about the 4th century BC. This ceremony went on for 2,000 years until it was profaned about 400 AD. But this was sort of the super bar mitzvah. Anybody in ancient Greece would participate for 2,000 years. I mean, that's much longer than modern civilization has been around. So the components of kykeon are also unknown. But mushrooms grew in the Middle East. They grew in Greece. So it's quite possible that mushrooms also were involved as part of the kykeon. And again, why would they be sharing mushrooms? You know, not as a dietary thing. Here, have this mushroom, have it for dinner. So it's more likely it had some sacred connotation. And I'm actually in contact with a fellow now who's doing research trying to figure out what are the things that could have been in the kykeon. So obviously, if you took mushrooms and mixed them with Syrian rue, for example, it would amplify the effect. So maybe the mushrooms also were involved in kykeon. So that's a very ancient use that goes way back in history. We know a lot about ayahuasca. Everybody in here probably has heard of ayahuasca. In ayahuasca, you have a mixture of two plants that are boiled together to extract the active components. So we have Psychotria viridis, for example, chacruna, has DMT in it, and Banisteriopsis caapi in the bark has beta-carbolines. DMT is not active orally. If you take it orally, nothing happens. It's broken down by monoamine oxidase in the liver. But beta-carbolines in Banisteriopsis caapi reversibly inhibit the monoamine oxidase that breaks the DMT down. So if you put those two together and boil them and extract out the DMT from the Psychotria and the beta-carbolines, you get this ayahuasca, which is an orally active form of DMT. Maybe I have to aim this at the computer. In the American Southwest and northern Mexico, we have the peyote cactus, Lophophora williamsii. It's used as a sacrament by the Native American Church. In the United States, they have protection under the Religious Freedom Restoration Act to use it in their ceremonies. So if you're a member of the Native American Church, you can use it, but if you're not a member of the Native American Church, you can be arrested for possessing a controlled substance. Now, in all these cases I've talked about, we have the religious connotation, the ancient ceremony in Soma, the vision of gods, the kykeon in the Eleusinian Mysteries, this profound sacred mystery. The ayahuasca used as a sacrament in South American churches, syncretic churches like the União de Vegetal or the Santo Daime. And then we have the Native American Church, where peyote is used as a sacrament. So this definition of these drugs that produce religious experiences is a very apt description of how these things have been used way back in antiquity. I don't know if this thing needs a new battery or what. Okay. And mescaline is in peyote. It's one of the least active psychedelics. The dose of mescaline is 200 to 400 or 500 milligrams. But it's also a classic psychedelic that acts by the same mechanism as DMT and psilocybin. I think the battery must be wearing out in this remote. Huh? You charged it all night? I've sucked the energy out of it. And finally, let me talk about these compounds. So on the right, we have Turbina corymbosus. It's a species of morning glory from South America, known as ololuki by the Aztecs. Presumably was used, the seeds were used when they couldn't get the mushrooms. And we have Hawaiian baby wood rose, which doesn't have a historical use, but it's a member of the morning glory family. It also has the same components. And then we have in the middle an ergot-infected rye head. And ergot will infect rye and grain products in times when the fall is cold and damp. And we had scourges of ergotism during the Middle Ages. People would eat bread that had been made with this ergot-infected grains and would have symptoms of severe seizures and disorientation, hallucinations, and profound vasoconstriction, meaning it would constrict the blood vessels in the fingers and toes to the point where they would become necrotic. They didn't get enough oxygen, and they would die. And these people would have, their fingertips and toes would be blackened as if they'd been charred in a fire. This is called St. Anthony's fire. And St. Anthony was the patron saint of people who suffered from ergotism. And these little black things, these little kernels, those are the actual ergot kernels. They're the sclerotia, or the hibernating form of the ergot fungus. And if you pick those off and put them in a medium where they can grow, they'll actually grow little tiny fruiting bodies, little tiny mushrooms. So like any other fungus. Now what these all have in common is lysergamides. So lysergamides are derivatives of lysergic acid. This is these 4 rings on the bottom hooked up to the C with a double bonded O and up to the nitrogen. If you take off that N at the top and put an OH there, you have lysergic acid. So most of the ergot alkaloids are derivatives of lysergic acid, where that N group and these R groups attached are different complex peptides. If those R groups are hydrogens, you have lysergamide itself, or ergine, which is what's in the morning glories. And it's a weak psychoactive psychedelic. It's not as potent as, say, LSD, but it is active. So if you grind up these morning glory seeds and make an infusion, and many people, probably even some in here, have done that, you will get intoxication. LSD, the diethylamide, those R groups are 2 carbon ethyl groups that are attached, and that is by far the most potent and profound of the psychedelics that we have today. And whenever I talk about LSD, I like to point out something. LSD was discovered by accident by Albert Hofmann in 1943. Most of you probably know the classic story. And if you had a child who suffered from some mental illness in the 1940s, you were the parent, you were blamed. Psychiatry at that era was all focused on psychoanalytic processes. So if your child had some mental disorder, something had failed in your parenting. You had been a dysfunctional parent, and you had women called refrigerator mothers because they were blamed for being cold and distant and had not nourished their children. So that's the reason your child was sick. So you had all these people going around with sick children believing it was their fault because they'd been bad parents. But actually, okay, whoops. But actually in 1953, Betty Twarog detected serotonin in mammalian brain. Now most of the serotonin in your body is actually in the gut, in the intestines, in enterochromaffin cells. It's used for mobility, enhanced motility of your gut and so forth. So Betty Twarog had found this little assay with an edible mussel called a quahog. If you hang this mussel up in an organ bath and bathe it in artificial physiological fluid, and you squirt serotonin in there, that muscle would contract. And the more serotonin was in there, the more it would contract. And so you could plot concentration of serotonin versus degree of contraction And using standard solutions of serotonin, you could estimate how much serotonin was in that particular tissue. So she was assaying different tissues, and she was working in the laboratory of Irwin Page at the Cleveland Clinic. And she said, I want to analyze the brain. And he poo-pooed the idea. There's no serotonin in the brain. Everybody knows it's in the blood, it's in the gut, so there's no point in doing it. She did it anyway, and sure enough, she detected serotonin. So if you look at the literature on serotonin prior to 1954, all you see are studies published about the role of serotonin in the gut and also in the blood clotting process. But after 1954, you start seeing these papers that are involved in serotonin in the brain. Where is serotonin in the brain? What's it doing in the brain? So there's a rapid increase in publication and interest in serotonin in the brain. And when you look at the structure of serotonin, you see that's really the sort of core part of LSD itself. They both have this tryptamine structure. Suddenly, people said, well, we know LSD is a really potent drug because by 1953, Sandoz was sending it out as an experimental psychosis-producing drug. So if LSD was doing that, and it looks so much like serotonin, maybe serotonin had something to do with behavior. So this was really the beginning of the recognition that neurochemistry had any role in behavior. Now, that seems strange today, but that's what they believe. So the discovery of LSD really catalyzed interest in serotonin and understanding that serotonin and neurochemistry played a role in behavior. So we could say that the discovery of LSD really initiated the beginning of serotonin neuroscience to a very large extent. Now, how do these psychedelics work? What they all have in common is they're agonist or partial agonist at the serotonin 2A receptor subtype. There are 15 subtypes of serotonin receptors, but it's the 2A subtype which seems to be the target for the psychedelics. They also will activate with equal potency the serotonin 2C receptor. We don't think that has any really relevance. And the tryptamines in LSD also activate the serotonin 1A receptor, and at least in animal studies, some compounds like 5-MeO-DMT actually have powerful effects on the 1A receptor. And I might point out that LSD is only a weak partial agonist. When we think about a receptor being activated by a transmitter, serotonin fully activates the 2A receptor. LSD only activates it to the extent of about 25 or 30%. So it's not even a full agonist, which makes its activity even a bit more remarkable. LSD does many other things. So we could say that the pharmacology of LSD is perhaps promiscuous or rich, but the 2A seems to be the key target. Now, there are arguments that other receptors, ancillary receptors, may influence the overall qualitative nature of LSD's effects, or maybe the other psychedelics, but the 2A is the key target that turns everything on. So this is a slide I added because my wife looked at my talk. She said, you're talking about receptors, and do these people know what receptors are? Many of you do. But I'm just going to mention, these are G-protein coupled receptors. This is an actual X-ray structure of a G-protein coupled receptor. This is its actual size, relative size. Let's see if I can get this. And so I've plotted the van der Waals surface of this receptor, and you can see approximately where the membrane would be. And these are the GTP binding proteins that couple, the G proteins that bind. So these are G-protein coupled receptors, GPCRs. And you can see the inside of this thing, the G-protein binding part of it, is actually the biggest part. That's the business end of this. People often think these receptors are big and something happens. The receptor is just something that allows a signal to be transmitted through the membrane to activate these G-proteins on the inside. So when a drug binds to the exterior surface, so if you look at that picture, the exterior part of the receptor is on the top. That's outside the neuron. So LSD would interact with the top of that receptor about a third of the way down and bind to the binding site. And it changes the shape of that protein. So it rearranges itself to accommodate the drug that's bound, serotonin or LSD or psilocin, whatever. And then the interior parts of the receptor, down inside, they change their shape. And that changes its affinity for the G proteins. So there's a G alpha, G beta, and G gamma. And the G alpha is the key one here. It has GDP bound normally when it's hooked to the receptor, guanosine diphosphate. And when the drug binds to it, it extrudes the GDP and picks up GTP and dissociates. And so these G proteins actually create the signals that happen inside the neuron. This is a simple schematic. These G protein coupled receptors represent 7 transmembrane helices. So they're long proteins that form stretches, that form alpha helices, like the screw or like a spring, and they weave in and out of the membrane and pack together in this bundle. So when the drug binds near the top of the receptor, it changes the shape, and they can interact with these G proteins inside. So there are GI, GQ, there's GS, there's 14 different G alphas, and they have a beta gamma, and they can also signal. In addition, there are receptor kinases, G protein receptor kinases, or GERCs, which can phosphorylate the serines and threonines that are inside the membrane and can recruit a signaling molecule called arrestin. So these G protein coupled receptors can create a variety of signals, and that's one of the things that makes understanding how psychedelics work a little bit complicated, because receptors don't just turn on and off. When they turn on, there are a host of things they can couple to, and what they couple to depends upon the drug that actually binds. It's called ligand bias or functional selectivity. So if you imagine serotonin binding to this receptor, it's a small flexible molecule. It gets down the receptor accommodates itself, it forms a complementary shape, and a certain thing happens. But now you have a big tetracyclic rigid molecule like LSD. It gets in there, and the receptor collapses around it, but you get a different shape altogether. And that changes what happens inside the cell. So the nature of the signals is different. So you get a variety of G proteins, GI, G-alpha, GS, or arrestin. It depends upon the ligand, so it's dependent on the ligand. So even though all these psychedelics are serotonin 2A agonists, they may generate different signals. So psilocin, mescaline, LSD, DMT may generate a different complement of signals within the neuron. So it makes the study of these at that level fairly complex. So where are these receptors? We know they have pretty powerful effects. So are they located in areas that would make sense? So this is a graduate student that failed out of the program. We sliced his brain across like this. And when you use a radioactive drug that binds to the 2A receptor, like ketanserin, I had a green pointer, but I was told that it wouldn't show up for the people that were doing this online, so I have to use this magnifier. Ketanserin, and if you look at this, you see that the color is related to the density of the staining. So after you soak the tissue in the radioactive ketanserin, you can lay it on a photographic film back in the old days, which is how this was done. And then after it develops, you can see where the high density levels are, and you relate it to the density here, relative density based on binding. So what you see is the serotonin 2A receptor is in all these outer areas of the cortex, the outer part of the brain, the most highly evolved and important part of mammalian brains. And those are expressed on apical dendrites of cortical cells in layer 5. So the cortex is made up of 6 layers. This is the exterior surface of the brain. Then layer 1 through 6 as we go down. And here's layer 5. And here are the pyramidal cells. And they're called pyramidal cells because they're shaped like little pyramids. They're pyramidal cells. And the apical dendrites are the parts of these that are just above in layer 5. And what you can see here, the blue lines represent inhibitory input into those cortical cells, and the red ones represent excitatory. So the cortical pyramidal cells in the cortex, all throughout the cortex, they're really the functional part of the brain. They're sort of like the central processing units. So all the information that comes in from all of our senses that's screened and sent to the cortex, it all gets sent to these cortical pyramidal cells, and they integrate this information and create a signal and send it back. So our higher thinking, everything is related to what these cortical pyramidal cells do. Serotonin 2A receptors are on those apical dendrites that modulate the sensitivity or membrane potential of those cortical cells. So since these are so important to function, if you start throwing serotonin 2A agonists in there and changing the membrane potential, you're not going to get normal effects. You're going to get something different. So now I'm going to switch. That's the background. I'll switch into what I call modern clinical research. I have to get a plug to the Hefter Research Institute because we started sponsoring a lot of these studies way before most anybody else did. And we've spent really a small amount compared with grants. It's a little over $7 million now, but all the money we get goes directly to the research. We don't have any overhead, we don't have any employees, we don't have any building, anything like that. So the money we get goes pretty much all to our research. And I'm not going to spend any time on these, but just to show you Charlie Grobe, who is here and gave a talk as one of the Hefter board members, published the first therapeutic study of psilocybin in 2011 in advanced cancer patients, showed that it had a salutary effect on their anxiety levels. Then everyone's familiar with the papers out of Johns Hopkins and New York University where, again, cancer patients were treated with psilocybin-assisted therapy, and that was published in December 2016. There was a lot of press on that. That really got people thinking. If you go to the internet, quite often you'll see a blog that says mushrooms for depression. That's related really to these two original papers. Matt Johnson gave a talk about cigarette smoking, psilocybin-assisted therapy, and nicotine addiction, and alcoholism by Dr. Michael Bogenschutz. Both of those studies are ongoing and are being expanded. The alcoholism study is enrolling 100 subjects at New York University. Studies the effects of psychedelics on gene expression. My son actually got interested in this field. I tried to dissuade him, but he's done it anyway. And he's looked at regulation of gene expression and cellular regulation after psychedelics. We have a laboratory in Zurich that we support, the Hefter Zurich Laboratory, that's been directed by Dr. Franz Vollenweider. And his associate, Katrin Preller, is here. She'll give a talk right after me. So he's done a lot of work. If you look up in PubMed, you'll see a lot of the papers he's done on emotional regulation and effects of psychedelics on behavior. He's now planning to do a big study of psilocybin-assisted therapy and depression. Also in cocaine addiction, Peter Hendricks at University of Alabama is doing a study of psilocybin-assisted therapy for treatment of hardcore cocaine addiction. And I'll show you a slide about that. And then effects on meditators, long-term meditators, that's ongoing. And we've got some others. We've got an opioid study, a safety study that's just starting at University of Wisconsin, and an obsessive-compulsive disorder study, and there's plans to do eating disorders. So we're trying to discover the range of possibilities. What can psilocybin-assisted therapy actually accomplish that we can't do with current pharmacotherapy? This is a study of cocaine addiction. He looked at the first 10 subjects, and this is an average of those 10. And basically, the psilocybin treatment is his top line. And from days 90 to 180 after treatment, you have 100% abstinence from cocaine. And these are averages. Some did better than others, but he has some really amazing stories of one cocaine addict who basically was living under a bridge and was homeless. And after this therapy, she got into social services, went back, got a housing, and started her life and said, you know, you saved my life. So these are really dramatic things that we've never seen with any other type of therapy. And then there's a lot of talk about the mystical experience, how important that is for the best outcome. And the nicotine study that Matt Johnson did, they published a paper, a follow-up paper that said the best outcomes were in people who had this mystical or peak experience. But then we had this paper that Ken Alper just published looking at ethanol preference in a strain of mice that prefer ethanol to water. He gave them a single injection of LSD, and for 46 days afterward, which is as long as he followed them, their alcohol consumption was significantly reduced. So you don't need the peak experience. Well, at least I don't think rats are having or mice are having a peak experience. So there's something physiological that's happening that goes beyond the psychology. So if psychedelic-assisted therapy works, how does it work? Because the conventional wisdom is that you have pharmacotherapy. So if you're depressed, they'll give you a selective serotonin reuptake inhibitor or an SNRI or bupropion. These are all related to monoamine transmission. It doesn't get to what's caused the depression. It's a palliative, and it doesn't work that well. You may know that SSRIs only work in about 50% of people when they work, and with therapy, maybe you get a little bit more. And that's a response. That's not actually a remission. A response just says you had a reduction in symptoms. A remission is the symptoms are gone. So SSRIs are not very effective. There's lots of them because that's all they have. If you have anxiety, they'll give you a benzodiazepine. So there are these targeted pharmacotherapies. Well, the serotonin 2A receptor isn't there's no targeted psychotherapy to that. It's a receptor that occurs throughout the cortex. It's not specific to any particular organ. So how is this working? How do these things have, an addiction? What's going on? So that's what I'm going to try to explain as best I can at this point. So Matt Johnson and my son and I wrote this paper, Psychedelics as Medicines, an Emerging New Paradigm. And we parroted a lot of the stuff that's been done at Imperial College that Robin Card-Harris has led on. And we said, probably there's this reset mechanism, and it's a different paradigm. I call it an emerging new paradigm. I really believe we're on the verge of a new paradigm in psychiatry, because psychiatry is bereft of drugs that work. And all the major drug companies have quit making drugs for psychiatric purposes. They make biologics for, irritable bowel syndrome and for arthritis and for cancer, but they've given up on the psychiatric drugs because they can't validate the targets, they don't know what to do, and there's nothing that really works. So this is a new paradigm that we're seeing, I think, and it's really exciting to be alive when there's this emergent new paradigm, because how many of us have ever seen a paradigm shift? And I think that's what we're seeing. I believe that's what we're seeing. So we talked about the possible mechanisms for that, and I'll explain a little bit of that as we go along. So this is a brain that was recorded using magnetoencephalography. It's a powerful technique that uses superconducting cells to pick up the currents that are produced in the brain. So when you think about a brain firing, maybe you think about neurons firing and a couple of neurons firing. That's not really what happens. The way neurons fire in the brain is through something called neuronal avalanches. And if you imagine an avalanche, how it starts at the top and then it picks up speed and it brings in more and more and more snow till you have this huge thing, neurons fire like that in the brain through neuronal avalanches. And how far and fast those avalanches spread in other neurons depends upon the sensitivity of the neurons that are involved. So you may have a branching of only one, where one neuron fires and it activates one other neuron. Nothing much happens. But you can have a situation where one neuron fires 2, and 2 fire 4, and 4 fire 8, and you have this avalanche. That's the way that the brain, how the neurons work. And so here's, if I can make this work, this is, you can see milliseconds, so this is real time. There's 1 second worth. So what you see are these spreading waves of depolarization that are producing currents that are picked up by magnetoencephalography. So this is how the brain is actually working. It's generating these currents and through this mechanism of neuronal avalanches. Now I tried to get a couple people that have done work with neuronal avalanches in vitro to actually put a psychedelic in there. They won't even answer my emails or phone calls for whatever reason. But I think this is the mechanism that underlies a lot of this firing of neurons in the brain. Okay. So that leads to this picture. This is actually, Amanda, this was in the back of the Beckley Foundation brochure. Work that came out of Card-Harris and Nutt Group at Imperial College that Amanda's been sponsoring. So when we look at a normal brain, there are anatomically defined areas. So you have hippocampus and amygdala and prefrontal cortex and parietal cortex and occipital cortex. You have these defined areas in the brain that talk to each other, and there's within those, let's call these areas nodes or hubs. So they're like little villages of neurons that just talk to each other and communicate what's happening on a day-to-day basis, And they don't usually reach out and talk to distant neuronal populations. But when you take a psychedelic, what you see is the integration in all those little nodes or hubs breaks down, and they all start reaching out and talking to each other. So it's kind of like a cacophony of, and Robin talks about entropy, increased chaos in the brain. So that happens, so now you see all these things that are talking to each other that weren't talking before. So intense global communication. And when the drug wears off and it drops back to the placebo condition, presumably it goes to a more healthy state. So every psychiatric condition that's known where they've done brain imaging using resting state functional connectivity, they found disruptions in that connectivity, which they call hub failure. And in at least one case where they treated people with an SSRI and they recovered from their depression, they found that their functional connectivity went toward a healthy state. So somehow there's a reset mechanism that occurs that when the global communication falls back to its non-drug state, those hub failures are somehow restored. And Robin Card-Harris has given a talk, and I've seen a couple of talks where they said, well, how does this work? And he shows this big red button with the white letters, reset. It's like the Control-Delete on a PC. Somehow there's a reset that occurs. Come on. You charged it all night? You go too far? Okay, so just to mention, and the Imperial College has taken the lead on a lot of this. So here's a paper for their treatment-resistant depression. They did brain imaging before and after, and I'm not going to read through this all, but basically what you see is there were changes in resting state functional connectivity in several areas of the brain that were correlated with positive outcomes in these treatment-resistant depression patients. And then, so then again, the conclusion is this process might be likened to a reset mechanism in which acute modular disintegration, these are these hubs or nodes where you have concentrated network activity, they're disintegrated, but then when you get global connectivity and they reconnect, there's some kind of a reset that's occurred. And that's what we don't know, how that actually happens. So now I'm going to try to get into how that might happen. We don't know clearly, but Agajanian did early work. He's at Yale University. He did all the early electrophysiology work with psychedelics, looking at cortical cells and what happens when you activate 2A receptors. So back in 1999, he was looking at cortical cells, and he punched tiny microelectrodes into cortical cells from rat cortex slices, and then applied serotonin or LSD to these cells to see what happened. And most of you know that neuronal firing depends upon a membrane potential across the membrane of the neuron. Neurons are, like electronic tubes, if you will, and they have a negative electronic potential inside. So, for example, the potential inside a neuron might be minus 70 millivolts with respect to the outside of the neuron. When the neuron fires, you have a depolarization. That negative potential goes back towards zero. You actually generate an action potential. But at lower concentrations of an agonist, you'll just see a depolarization. So maybe the membrane potential will go from minus 70 to minus 60 millivolts or minus 50. Until you get to the threshold potential and actually generate an action potential, they just generate postsynaptic currents, excitatory currents. So they found that when they were looking at superfusing serotonin onto these layer 5 pyramidal cells, they got what's called a synchronous release. They would see an excitatory postsynaptic potential right away. And then when they quit putting serotonin onto the pyramidal cells, when they continued to record, the pyramidal cells started firing in what's called an asynchronous mode. In other words, they were firing without the application of an external transmitter. Now, this was very puzzling. They couldn't figure out what it was. They thought, well, maybe when we stimulate the cortical cells, there's a retrograde transmitter of some kind that's activating neurons from the thalamus, and maybe they're releasing glutamate or something like that. They didn't really know. It went until 2007 when Baik and co-workers did a very controlled and systematic study trying to figure out, is there an anterograde transporter produced? Are these from terminals from the thalamus? Where is this glutamate coming from? They concluded the glutamate was coming from deeper layers of the cortex, under layer 5, and they were sending excitatory projections up and releasing glutamate onto these layer 5 cells, which that's what Agajanian was measuring. So then in 2016, David Martin and Charles Nichols, my son, they found a method to separate and purify cortical cells. So they took rats, they gave them DOI, which is a psychedelic phenethylamine, and they homogenized the cells, and they used a novel technique they developed called fluorescent cell sorting, where they could pick up the cells that express both serotonin 2A receptors and also cells that actually had generated action potentials. So when a neuron fires, there's a substance called CFOS, CFOS, which is an immediate early gene that turns on. So they said, let's get all the cells that have both serotonin 2A receptors and that produce CFOS. So they've been excited and actually generated action potentials. Because when you apply serotonin directly to layer 5 cells, they don't generate action potentials. They depolarize, but they don't generate action potentials. So they identified the cells, and what they found were there were areas deep in the cortex, which corresponded with what Bayek had said. There were cells that had 3 to 4 times the expression level of serotonin 2A receptors, but which also fired. And they identified those areas as being in the medial prefrontal cortex, the somatosensory cortex, orbital cortex, and the claustrum. So the claustrum is really interesting because Francis Crick and Christoph Koch wrote a paper in 2005 saying, is the claustrum the seat of consciousness? Because the claustrum is a very enigmatic structure in the brain that nobody has really studied very extensively. And this is what it looks like. It's the most highly connected area of the brain. It's between the cortex and the striatum. So these are projections out to the cortex. The claustrum has the highest density of serotonin 2A receptors in the brain. So there may be something to this. This may be a key organ where this is happening. And so Martin and Nichols proposed that this was a trigger population that excited these cells, which then sent glutamate excitatory projections out to the rest of the cortex and turned on what we see with these network changes. And I don't have the math to explain this to you, but this paper just came out a couple weeks ago. It's by Gustavo Deco, a really well-known brain imaging specialist in Barcelona, Spain. And what he did was you can actually use a type of diffusion MRI, and you can actually map out all the white matter tracks in the brain. White matter are the tracks where the neurons go. He could use the fMRI data from the Card-Harris experiments, which showed the dynamics, brain dynamics, and then he got high-resolution mapping for several receptors that have been done by Gideon Knudsen in Copenhagen, looking at 2A receptors, 1A receptors, 5-HT1B receptors, 5-HT4 receptors, and serotonin reuptake carriers. And then parcellated the whole brain into 90 different cortical and subcortical structures and put them together in a whole brain model. And like I say, I can't explain the math of this, but got a whole brain model. And what they also found was that this only correlated with the density of 5-HT2A receptors. So this again was a proof that the 2A receptor was involved, because if they used the 1A receptor or the 1B or the 4 or the uptake carrier, nothing correlated with the dynamics that they were seeing, as well as the 2A receptor. So these psychedelics have turned out to be really important tools for understanding brain function. So I'm pushing my time here. So the other thing I'd like to talk about is what happens with dendritic spines. So we know that in depression, in post-mortem brains of people who are severely depressed, in the hippocampus, there's decreased spine density. And spines are areas that have receptors on neurons. So there's a decreased spine density. And loss of hippocampal neuroplasticity is also related to depression. So in 1997, a group had shown that if you look at 5-HT2A receptors, they actually modulate the formation of brain-derived neurotropic factor, BDNF, which is a key growth factor for spine neurogenesis and synaptogenesis in the brain. And in rat primary cortex cultures, stimulation of 5-HT2A receptors by DOI caused a transient increase in dendritic spine enlargement. And without spending a lot of time on this, the way spines work is they have a type of glutamate receptor called an AMPA receptor. And if glutamate hits those, it causes a maturation of the spines. They grow and spread on AMPA receptors. And so this is something that is very important for normal cognition. I'm almost just about out of time. So let me just mention there's a whole biochemical pathway, and I'm not going to explain this, but glutamate hits AMPA receptors, and that leads to the membrane change, leads to voltage-dependent calcium channels. That releases calcium. And you have BDNF produced, which activates its receptor, which is a TrkB receptor. And there's a whole complex series of events that lead to expression of more glutamate receptors and spinogenesis, increased growth of spines. So that happens in response to serotonin 2A receptor stimulation. And this is a paper that came out of David Olson's lab this year. showing exactly what's going on there. So here are looking at spines. Here's with vehicle, no treatment at all, DOI, DMT, and LSD. And when you stain for spines, what you see is the growth of spines. You don't see that in the vehicle. Come on. You don't see that in the vehicle-treated animals, but you do see it with DOI and with DMT and LSD, which is the most potent in stimulating spinogenesis, neurogenesis. And I know I'm out of time, but another minute or two. So I just want to add one other thing. Something my son discovered quite accidentally is the anti-inflammatory effects of psychedelics. There's a lot of interest in the psychiatric community now in neuroinflammation. Inflammatory processes in the brain are thought to be responsible for certain types of psychiatric psychopathology. So what he discovered accidentally was that psychedelics are potent anti-inflammatories. And at least, this is when rat aortic arch they used. These are pro-inflammatory factors, so ICAM1, MCP1, and interleukin 1B. All of these are significantly attenuated after you apply TNF-alpha. So TNF-alpha is an inflammatory. If you look at some of the biologics for treating arthritis like Humira, it blocks the effects of TNF-alpha. Well, serotonin 2 agonists block the effects of TNF-alpha, and they're very potent. So in the aortic arch, and this is asthma, he's shown that you can reverse or prevent asthma in a mouse model of asthma. So the way they do this is take mice, they spray an aerosol of albumin up their nose. So here's a normal mouse, and this is the way it looks. It has no inflammatory cells. This is a cross-section of the airway. And if you spray OVA in their nose, eventually they become sensitized. This red is mucus production. There are also inflammatory cells produced. But if you give DOI along with the OVA, you don't get that effect. And they've actually taken mice, have developed this asthma model, given them DOI, and can reverse it. So in addition to this change in neurogenesis or spinogenesis, there may also be anti-inflammatory effects that are happening. All the psychedelics are anti-inflammatory and much more potent than typical things like ibuprofen or naproxen. Extremely potent anti-inflammatories. So this may be another thing that adds to the therapeutic effects. So I only ran 2 minutes over, so thank you.
David, for sharing your knowledge with us in your very informative talk. And because you kept your time very well, we have 15 minutes for questions. So if you have questions, there is a microphone on the table here. Come up front, please, and you have the unique opportunity to ask David.
Thank you for a fascinating talk. And I'd like to ask two questions, and one is about hypothalamus that is central for regulation of moods, like peptides, vasopressin, oxytocin, and so on. And what about serotonin and LSD regarding hypothalamus? That's one question. The other, is there also a cell destruction effect of LSD coming into these synapses?
So the second question, is there a destructive effect of LSD? Not so far as we know. And in fact, in the studies published out of David Olson's lab, you actually have increased neurogenesis and nerve growth. So just the opposite.
But couldn't this increase be a repair activity?
So he's taken normal mice and given them LSD. So there's no repair. I mean, they're not damaged to begin with, so it's a stimulation. And this has been shown by other investigators looking at in vitro systems. If you activate 2A receptors in a number of systems, it'll stimulate spinogenesis and neurogenesis. So if you consider that depression is related to the loss of spine density and decreased neurogenesis, then what we're doing is we have a process that reverses what we see in depression. Now, that may not actually, it might be a red herring, but as far as we know now, it looks like psychedelics will stimulate neurogenesis as well as possibly be anti-inflammatory.
Hi, you mentioned that someone's going to do research on psychedelics and eating disorders. Could you tell me a little bit more about this?
That protocol is not written. When I started the Hefter Institute, Mark Geyer and I met Franz Wollenweiter in Switzerland, and that was the first thing we wanted to try, because the eating disorders, as you know, are the most lethal of all psychiatric disorders. They're not treated well at all. We don't actually know how many, mostly young women, die of eating disorders, because often on the death certificates it said they died of cardiac failure, but they don't say secondary to severe anorexia. So we've been interested for a long time. The problem with being a not-for-profit is it depends on philanthropists. And so philanthropists have said, oh, we want to study depression, we want to study anxiety. So we have a protocol that's nearly ready. We're going out, and this is one of the things on our wish list that we send out. We want to do eating disorders. We have anecdotal reports of women who have said, you know, I ate mushrooms and my eating disorder went away. We have a couple. So I think there's a signal there, and it's in the compulsive disorder spectrum, OCD. So I'm very interested, and if you know anybody that wants to spend a couple $100,000, we would sure push that protocol up.
I'll figure something out, okay. Thank you very much.
Hi, thank you very much for a very interesting talk. I was particularly interested in this anti-inflammatory approach. To your knowledge, do you know if there's any studies on psychedelic effects on the effect of psychedelic compounds on peripheral biomarkers, like plasma biomarkers, like CRP or interleukin 6, or any studies at all?
I'm not aware of any. The things that my son has done in vitro are with rat aortic strips. And actually he discovered this because he was working, they had a model for atherosclerosis where they would inject TNF-alpha into these aortic strips and they would have inflammatory marks would be generated. So in theory, based on what he saw, people could be taking things like psychedelics and would be blocking development of atherosclerosis. But nobody, there's no money for that. He's sent grant proposals in, and they go into study sections of people that are looking at biologic factors, and they say, what? Psychedelic is an anti-inflammatory? And it's good data, but the study sections are not ready to, they have people who are invested in looking at all these biomarkers and biologics and things. So the idea this could happen is, and he's got additional data. He's got a whole library. He's doing a principal components analysis to determine actually how that happens. I think eventually that'll get done, but there's just no money for this. Studying psychedelics has been, a wasteland financially for a long time. It was a kiss of death if you were an academic, except for a few people.
So let's hope to see more of those studies in the future.
I mean, it's really exciting. He's got more papers that are going to be coming out, but as far as I know, he's the only one that's looking at that.
Thank you very much.
Thank you for an amazing talk. My question is about psychedelics and psychotic illnesses. You told that the decrease of default network activity could be related to the beneficial effects by first increasing chaos, entropy in the central nervous system, and then taking it gradually into reset state. How would that relate to psychotic states or illnesses?
You mean like schizophrenia?
Yeah, for example, in treatment or modeling, or do you have any ideas about that topic?
Stan Grof, if you've read his books, he talks about treating schizophrenics with high doses, massive doses of LSD. And he was actually ostracized from the psychiatric community because of these studies. And he, in his books, I think it's Realms of the Human Unconscious may be the one. He has two major books, but in one he talks about treating people with schizophrenia with high doses of LSD and taking them back and regressing them back through these layers of this coex, and then getting them to a point where they're sort of connect with the in utero. And then he says they were medication free. Now, I haven't seen scientific studies of that. I find it kind of incredible. But it's possible that maybe there's some kind of adaptive neural remodeling that would happen. But I think schizophrenia is considered to be a sort of developmental disorder where you have a death of white matter in the cortex that occurs over a period of time. So I don't know whether you could reverse that with a psychedelic. You'd probably have to do it early on before it progresses. I think they can pick up prodromal evidence of developing schizophrenia at age as early as 13, but basically you don't see it till late adolescence or early adulthood.
Yeah, and it's also exclusion criteria for most of psychedelic studies, so it's really hard to get any data addressing this question. But yeah, thank you.
Hello. You say we are on the verge of a new paradigm in psychiatry. Could you elaborate a bit?
Well, I think the current paradigm is directed pharmacotherapy. You have an illness, okay, schizophrenia, so now we're going to give dopamine receptor blockers, or atypicals block D2 receptors and 5-HT2A receptors, or for depression, you're going to use an uptake blocker. So the current paradigm, which doesn't work very well, is targeted pharmacotherapy based on what the illness is. So now we're looking at a change in paradigm. We're not doing targeted pharmacotherapy anymore. We're saying there's something different. We're looking at the overall brain. And what I used in this paper, Psychedelics as Medicines, we used an analogy that suppose you have an electronic circuit board for your stereo or whatever, and it's not working. And you don't know anything about electronics, and you want to make it work. What would you do? So maybe you'd go in, you'd replace this transistor and see if that fixes it. And you'd replace this inductor and see if that fixes it. Replace this resistor and see if that fixes it. That's kind of like what psychiatrists do with depression. Try an SSRI, try an SNRI, try bupropion, and maybe you get the ECT. It's a trial and error empirical approach. And so we're saying, that's a completely different thing. That's kind of a trial and error approach, and now we have this idea of network connectivity, restoring it to functional health. So that's the shift in paradigm, looking at these illnesses and treating them in a different way rather than focusing on a targeted pharmacotherapy. So that's kind of what we meant.
Thank you.
Sure.
Hi, thank you for a great talk. I was really fascinated by the slide about the Claustrum, and that you mentioned the studies of Christoph Koch and Francis Crick. Do you think there will be more studies with psychedelics in regard to the study of consciousness within that field in relation to, or in like a sidetrack to psychiatry?
Yeah. After that paper appeared in 2005, there was a whole flurry of publications, and there's actually now a Society for the Study of the Claustrum. Christoph Koch was not interested in psychedelics for a long time, but he has recently, I understand, become interested in how those affect brain dynamics. And Franz Vollenweider has interacted with him because Franz has been doing studies with psychedelics, so they become close colleagues. So I think the study of psychedelics is going to lead to changes in how we understand brain function, like this paper that Gustavo Deco published. He's a well-known brain imager in Barcelona, has published like 400 or 500 papers. So when people of that caliber start saying, we can use a psychedelic to put this whole brain image together, he points out, we can block the effects of a psychedelic with ketanserin, which is a 2A antagonist. And so that's been you can block it in humans and animals, and you can block psilocybin and LSD with ketanserin. But it was interesting, he looked at these high-resolution receptor density maps from Getty-Knudsen's group, and he had the 2A map, the 1A map, the 1B map, the 4 map, and the CERT map. And he replaced them all to see, and only the 2A map really was consistent with the dynamics they were seeing. So these kinds of ideas are going to lead to, I think, a much better understanding of psychopathologies when they have maps of different receptors and they look at a disturbed state in someone with schizophrenia, for example, and look at these different maps and say, okay, here's where we see the problem. So I think the psychedelics are going to be tools in helping us to understand that, but it's going to lead to changes. I mean, this whole idea of brain dynamics has really been growing rapidly in the last, say, 15 years. So I think it's going to help. It's going to be one of the tools that help us understand consciousness a lot better.
Thank you very much. Also, I was interested in the first question about correlation between psychedelics and the hippocampus and the amygdala. Is there any studies on that?
There are some with the Carr-Harris study with treatment-resistant depression, the parahippocampal connectivity with parahippocampus and prefrontal cortex. So parahippocampus is involved. There are some changes there with depression, but they really aren't enough detailed maps. I mean, in the studies that have been done prior to this one by Deco et al., they've only looked at 10 or 12 different areas, where he looked at 90 different parcellated into 90 different areas. So the computational power has been lacking till recently for doing these kinds of studies where you combine these high resolution maps of all these things together. So
Thank you very much.